Hi my dear students, welcome back to new session. In the last class we already learned regarding vaccination, immunization, autoimmunity and lymphatic system of a human you had learned in the last classes. The today class is regarding AIDS. In NCERT, two chronic diseases are given. One is AIDS, second one is a cancer. Well-known information that is regarding AIDS. Acquired immunodeficiency syndrome. The literal meaning of AIDS is deficiency of immunity system that acquired during the lifetime of an individual. It is indicating that it is not a congenital disease. Alright? Let's write the points here. AIDS means the word is acquired immunodeficiency syndrome. Right? The literal meaning of AIDS is it is a deficiency of immunity. It is a deficiency of immunity that acquired during the lifetime, right? Lifetime of an individual, right? And it is indicating that it is not a congenital disease. It is indicating that it is not a congenital disease. Right? What is the main congenital disease? It means simple birth defects. It means what? Birth defect. For example, the babies are like inborn babies suffering from trisomic condition or heart diseases or physically or intellectual powers are very less. So that we can call as what? Congenital disease. Simple birth defects. Right? So the AIDS is not a birth defect. Right? Here the birth defects is generally we can give an example. For example, heart problems, heart defects or Down syndrome or sometimes cleft or broken lips or cleft lips, as well as sometimes physically or intellectually weak, right? So in such case we can call it that is congenital disease like trisomy conditions. And here, for example, in case of if the disease is transferred from mother to fetus, inborn baby, so in case we can call as it is congenital HIV. If the HIV virus, it is transferred from contaminated mother, nothing but AIDS mother to fetus. In such case, we can consider what it is congenital HIV, right? So here, generally, so this HIV, it is beginning in 1981. It was first noticed in one homosexual male. AIDS, it was first recognized in homosexual male in USA in 1981. At the beginning, they given different names of the virus. They not use the HIV virus name. They not use it. Actually, HIV virus, the name is used in 1986. At the beginning, they used various names. The various names are HCLV3 or HTLV3, and third one is LAV, and the last user name is HIV in 1986. The first one HCLV, it means human cell leukocytic virus. Human cell leukocytic virus 3, they used various names are used at the beginning for AIDS. Later, human T lymphocyte virus. Human T lymphocyte cell virus. Later, lymphocyte associated virus. At the last, in the 1986, they used one name, that is human immunodeficiency virus. Right? So we can guess one question from here. Which of the following, like one multiple choice question, right? Human cell leukocyte, human cell leukocyte virus 3, right? Second one is what? Here, lymphocyte, human lymphocyte, human T lymphocyte virus. Next, lymphocyte associated virus. But the last that used the name in the 1986, that is HIV, human immunodeficiency virus. This is nowadays we are using this name, right? Human immunodeficiency virus. From till 1986 we are using this name, right? So this is regarding HIV here. So later within the 22 years, it is spread throughout the world. And within these 20 years, nearly 25 million people are died due to the AIDS. Now, after 1986, within the 20 years, it is spread throughout the worldwide. Worldwide it is spread. And due to the AIDS, due to the immune deficiency, nearly the number is little bit different in different books. 25 millions of people are died due to this AIDS, right? Next, here HIV is caused by which one? Sorry, AIDS. AIDS, the disease which is caused by virus. The virus name is what? HIV virus. Okay. Then let's focus on some information regarding HIV virus. HIV virus. So these 2 points you already know that, right? So virus is what? A virus is when it is present outside of the cell, it is equal to one dust particle. So it have no capacity to infect. When it is entered into the human, or when it is entered into the one host cell, then it has the capacity to replicate itself and interfere in metabolism of an individual, right? So these are the 2 points. Next, and one more point. Generally, viruses are 2 types. One is plant viruses and animal viruses, but this one is an exception. So generally, plant viruses are containing what? RNA. Animal viruses are containing DNA. Here, this HIV virus is causing disease to the human. We can consider as animal virus. But exception is here. Here, this animal virus do not contain genetic information as DNA. So in this case, genetic information is not a DNA like other animal viruses. Okay. So let's see some structure of HIV. HIV virus, right? HIV virus. So this HIV virus, which will belong to one broad group, that is erythroviruses. As usual, the virus is containing the first outer coat is protein, that nothing but capsid, and it is containing the genome. The HIV virus genome is genetic material is RNA. Two single stranded RNA molecules are present in the genome of HIV virus. And along with the number of enzymes are there, but especially three main enzymes are present. Out of three, very important enzyme is a reverse transcriptase enzyme. A reverse transcriptase enzyme, protease enzyme, and an integrate enzyme. These three enzymes, two single stranded RNAs, and outer coat is a capsid coat. So along with this capsid coat, it is also containing some small spikes are present on outer surface, such are made up of glycoproteins. Such are made up of what? Glycoproteins. And here inner protein coat proteins are different and outer protein proteins also be different. Inner protein coats are made up of like a P28 and outer is made up of like P18. Okay. So this is information regarding inner is, let's write the points, right? HIV is belong to one broad group, that is retroviruses. Retrovirus. It is one of the broad group. And the virus is, it is made up of, like as well, the outermost layer is protein coat. Protein coat that we can call as normally capsid. We can call it what? Capsid. So the capsid is containing spikes of proteins. Spikes of proteins. So which are complementary to antigen receptors of different cells in the human, right? Which are complementary to receptors, antigens here, receptors of cell surface. Especially these cells are, example, the cells are macrophages, lymphocytes, dendrons, like this one. Here, what I am telling here the outer coat is containing spike-like structure, which are complementary to receptors of other cell surface like macrophages, lymphocytes as well as dendrites. Okay. And here, if you observe the genome of HIV virus, the genome is containing what? Two single stranded RNA molecules are present, right? And along with these two single stranded RNA molecules, enzymes. Here number of enzymes are there, but very best example are important enzymes are first we have the key reverse transcriptase. Just recall the knowledge we discussed this reverse transcriptase in the protein synthesis. Central dogma of reverse transcriptase, the method is proposed by Teminism, right? So first enzyme is reverse transcriptase enzyme, second one is protease enzyme, and third one is integrate enzyme. So these three enzymes, two single stranded RNAs and cell coat, protein coat, right? Next, if you observe here, proteins are protein coat. We can divide into two types. One is inner side and outer side. Proteins. Inner side proteins are made of P24. Outer side is P18. Right? So this is actually inner side coat, outer side coat. Okay. Then if you observe the diagram, like the diagram is, this is one HIV virus. Here, orders is a lipid layer. Lipid bilayer. Okay. And here, like spikes are present here, spikes. Like spikes are present. Like spikes are present around the complete virus, right? So these are glycoproteins. These are glycoproteins. Okay. And next, here, this is the inner side of protein. Inner side of protein code. Here, this single stranded, here one RNA, and here also the one single stranded RNA. One enzyme, second and third. Maybe this one is ssRNA, single stranded RNA, and one is a reverse transcriptase enzyme, and one is integrase enzyme, and this one is protease enzyme. Protease enzyme. And here this inner code which is made up of P24 now, P24, and here this is outer set of proteins. There will also be outer set of proteins are there. These all are so which one made up of P18 proteins. Okay, so this is actually structure of HIV virus. Next, transmission of HIV. Transmission of HIV. Here, the disease is spread through first, most of the cases here, sexual contact with the infected person. That is the first reason. And second, those are required, repeated blood transfusion is required, right? That is the second. And third one is, so those are taking drugs, intravenous drugs, and abuse persons. They are sharing their needles, this injection to one person to another person. So this is actually main. And again, like blades in barber shops. These are the normal transmissions. The first one is sex contact with the infected persons. That is the first. And second, those who are need repeated blood transform, right? And here during the organ transplantation and during the transfer of blood in case of contaminated blood is injected in during the blood emergency, right? And normal also you can write, for example, normal blades, razors in barber shops. So these are the main reasons of transmission of HIV to one person to another persons. So in high risk, mainly high risk of transmission in case of those who are maintaining multiple sex partners. Those who are maintaining multiple sex partners, here they face a high risk of transmission of HIV disease first. And second here, so here infected mother. So here the disease is transferred from mother to fetus through placenta. So here the baby also going to suffer here, right? And third one is repeated blood transfusion or repeated blood need. In NCERT they are asking the question is, so in which case, then which persons are needed a repeated blood transfusion? Those who are need the repeated blood transfusion, which are suffering from blood disorders, diseases. You know that one is sickle cell anemia, thalassemia, right? Sickle cell anemia, right? Thalassemia, and it is also blood cancer, right? So these persons are needed repeatedly, especially thalassemia persons and sickle cell anemia and blood cancer. They are repeatedly every 1 month, 2 months they are required. Fresh blood is needed, right? So here these are facing high risk. Next one is misconceptions. Misconception at the time of 1995. So most of the people believe that disease is spread by mosquitoes. Here, but the scientists have proved that the HIV virus is not transmitted through vectors or through virus, and it is not also be spreaded by through hugging, kissing, or shake hands, or like sneeze, cough, sharing of glands, sharing of object, utensils. So by this the disease is not going to transfer. Very clear, crystal clear. When the disease is transformed only in a fluid contact, nothing but vital contact, nothing but blood contact, body fluid contact. In such case only the disease is transferred to one person to another person, right? Misconceptions are the 1 or 2 mark question. Write it. Misconceptions are it is not transferred through vectors like mosquito, mosquitoes, and here the disease also be not spread or like sharing of like objects or cloth or sharing of food, saliva, as well as like which one, as well as shake hands, and like these all are, right? And hugging, kissing, these also be not going to spread the disease, right? So these are the misconceptions of HIV. Next, then virus replication within the human cell, right? Due to when it is infected, when blood transfusion case, and second one is mixing of body fluids. In such case only the HIV is transferred to one person to another person. Then it is entered to one person to another person. First it is going to infect the macrophages, lymphocytes, dendrites, and lymphatic systems. Then let's see how the virus is replicated within the host cell, right? Just put the heading replication of virus. Replication of virus in host cell. Replication of virus within the host cell. And already you know that the virus is made up of what? These 2 or 3 points. It is made up of like protein coat, that is capsid, that is containing 2 single standard RNA molecules, as well as mainly 3 enzymes: a reverse transcriptase enzyme, protease enzyme, integrate enzyme. These 3 enzymes are present in like virus genome. So let's see how the virus is going to replicate within the host cell, right? First, when it is entered into the blood, the first it is going to attack or attach to the macrophages. The question is here, sir, number of cells is there, then why it is always going to attach or infected to only macrophages? What is the reason? The reason is what? The reason is HIV, it is containing spike proteins which are complementary to receptors of macrophages. So that's why immediately it is going to attack or infect to the macrophages, and it is not going to kill the macrophage cell, just it is a comfortable location to replication of virus. Okay, let's see. The first virus is infect to macrophages. Why? Because the reason is what? The reason is the spike proteins are complementary to receptors of macrophages, right? So that's why it is going to first attack to only macrophages, right? Now through these receptors, this virus cell is enter into host cell. The host is what here? Now it is entered into the cell through endocytosis process. Through endocytosis it is entered into the macrophage. So during this, the protein code is shed outside of the cell. Only the genome is entered into the host cell, right? During this process, what happened? Proteins are shed outside the macrophage cell, and which are entered into the cell? Only genome means here what? 2 single stranded 2 ssRNAs as well as 3 enzymes. 3 enzymes are entered into the host cell, right? Next, when it is entered into the cell, immediately like first enzyme is activated, which is a reverse transcriptase enzyme is activated and production of DNA. So later it become as cDNA, we can call as complementary DNA. Later this DNA become as double stranded DNAs are formed with the help of which one? A reverse transcriptase enzyme. So during this reverse transcriptase enzyme, the chances is there to form number of mismatches of nucleotide. But here this mismatches of nucleotide sequence is not going to corrected by any type of enzymes, because in RNA DNA proofreading enzymes are absent. So that's why, these are errors. These errors are not rectified during this reverse transcription. Okay, but in our cells, in human cells, in eukaryotic cells, DNA proofreading enzymes are there. So that's why during the transcription or during the DNA replication, if errors are formed, such errors are corrected by DNA proofreading enzymes. If errors are formed, the cell is not going to allow the further cell divisions, right? So that is our system. But here what happened? Here, like DNA proofreading enzymes are absent. So that's why every time new nucleotide sequence is formed. So this new nucleotide sequence is going to code for new proteins. New proteins are formed, like new antigens are formed. So further, our immunity system is going to produce one antibody for one specific type of antigen. But later what happened? Later, due to these genetic errors, again new antigen is formed. For that, our body again going to produce new antibodies are produced. So that's why it is a time lagging process. We can't neutralize it immediately. This is a very tough task to human immunity system, right? Right at the point. When it is entered into the cell, first which one is a reverse transcriptase enzyme. First it is activated and produce from RNA to reverse transcription to form of cDNA. At the beginning we can call as complementary DNA. Later is become as dsDNA, double stranded DNA, right? During this process, number of errors of nucleotide sequence. We can notice number of errors of nucleotide sequence. So these errors are not rectified or not corrected. Not corrected. Why? Because DNA proofreading enzymes are absent in this HIV virus, right? So for that what happened? So for that every time of replication, it is going to produce new sequence of DNA is formed. So new sequence of DNA means what? It's going to code for new type of proteins, new antigens, and for that new antibodies have to produce. So that's why it is a tough task to control by human immunity system, right? Next, here, so this is actually a role of first enzyme, and second enzyme is what? Second enzyme is integrate enzyme. So now with the help of integrate enzyme, now this double stranded DNA is going to integrate it with a host genome nucleus containing chromosomes. We discussed in the biotechnology. So the DNA individually, it is not going to replicate itself in host cell. When it should be replicated? When it is a part of host genome, then only this foreign DNA molecules is going to replicate it. Apply the biotechnology knowledge here, right? So with the help of integrated enzyme, which one? That is dsDNA, double stranded DNA is integrated with a host genome. Host cell here what? Macrophage. Now macrophage DNA as well as viral DNA both are mixed. Now it is going to replicate itself. Now it replicate along with the macrophage genome. Now during this replication, number of particles are going to form. Number of DNA particles, number of virus particles are formed. Later, from this DNA, again transcription, translation is occurred, proteins are formed. In that protein, some proteins are become as glycoproteins, they made up of capsid, and some proteins is act as enzymes. So now what happened? After somewhat time, here you can see it will take some months. After somewhat months, glycoproteins are formed, and some enzymes also be formed, and number of time of replicated DNA information also be there. Then what happened? These all are going to assemble to form, and thousands of virus particles are formed within the cytoplasm of macrophage. Then later, when the number is increased, so these cells are off from the macrophage and again released into the blood by exocytosis process. Right? Right the point? Virus particles here, and again here transcription. Later translation is formed to form of proteins, and this protein, some proteins are act as what? Some proteins are useful made up of capsid, they become as capsid, protein coat, and some are become as what? Enzymes. Now here everything is there. Here capsid is there, enzymes is there, and like virus particles, genetic reform also be there. Now these all are going to assemble to form one complete virus particle. Nothing but HIV particles are formed in cytoplasm of macrophage cell, right? When the number is increased, automatically they are going to off from the macrophage. By process is exocytosis. Okay, so that's why we can known as this macrophages we can consider as it is a factory. It is a factory of HIV. It is a factory of HIV. Okay. Now through exocytosis process, the number of virus particles, HIV particles are off from the macrophages or entered into the blood. Right. At the beginning, these cells are called as what? Pro-virus. Pro-HIV virus particles. Here the DNA sequence is already changed in this daughter molecules, nothing but proviruses. So these changes are very useful to attach or infection of lymphocyte cells, that is helper T lymphocyte cells. Here the problem is came. Now here when they are interacted or attached with a TH lymphocytic cell, helper T lymphocyte cells. So these enzymes are going to affect on T lymphocyte cells, then gradually the T lymphocytes number is going to decrease. The number is going to decrease. Then what happen? If TH cell, we know that helper T lymphocyte cell number is decreased, then what happen? These helper cells are very necessary for stimulation of B lymphocyte cells. So further the B lymphocytes is going to produce antibodies, right? So here if T4 nothing but TH lymphocyte number is decreased means gradually antibody production also be decreased in that person. So antibodies number is decreased means what? Immunity system is become as weaker, nothing but deficiency of immunity system, right? So this is actually effect of HIV virus in human. Let's write the point. So through exocytosis, these virus particles are come out or off from macrophage cell, right? Now these are called as, such cells are called as proviruses. At the beginning they are called as nothing but daughter viruses. Here these are what? This DNA sequence, the nucleotide sequence is already changed. So which is useful to infection of TH lymphocyte cells. Again what happened? Again with the help of three enzymes, reverse transcriptase, integrase enzyme and protease enzyme, it is going to attack on T lymphocyte cell. But here due to that gradually this TH lymphocytes cells are gradually they are going to die. The number is hugely reduced. If TH1 number is decreased, what happened? So in such case B lymphocytes are not stimulated. So for that what happened? For that there is no production of antibodies, right? If there is no production of antibodies in the human, it means immune system is going to weaker. Immune system gradually what happened? Become as weaker. So this is actually the main character of HIV virus, right? So if immune system is weak, then what happen? If any simple pathogen is attacked, so our body is not going to respond, not going to counter action of that antigen. So gradually the human body is, you can see, like factory of number of pathogens, number of infections. The person is suffering from number of bacterial, viral and protozoan infections, right? After light you can see, then after somewhat years, due to this infection only the person is going to die, right? Next here symptoms or incubation. Next incubation time. So here for incubation time it is in 2 to 10 years. Here in some book is given 2 to 8 years, 2 to 5 years are given, but minimum is 2 years, maximum is 10 years, right? At the beginning, it means at the beginning 6 to 8 months at the time, symptoms are not appeared. Symptoms are not appear, right? So that stage we can call as a window stage. Window stage. That stage is called as what? Window stage. It means to turn the HIV infection to AIDS. So it is time taking process. Sometimes it take years. Sometimes it's take years also. Okay. After somewhat years of HIV infection, the person it become as AIDS disease is appeared. So this is regarding incubation time. Next, symptoms. What are the symptoms? So we know that already at the stage, at the beginning, it means 2 to 6 or 6 to 8 months, right? Symptoms are not appeared. Here, when Th lymphocyte cell number is gradually decreased, then only symptoms are appeared. Then only symptoms are appeared. When the immunity system is become weaker, then only symptoms are going to appear. Right? The symptoms are at the beginning normal symptoms. Normal symptoms are like fever, headache, little bit diarrhea. Then after window period here, ARC, AIDS, related characters. So S means what? Syndrome. Syndrome means what here? Number of group of character, not specific particular one character. Group of characters, right? So ARC we can call. When we ARC here, the gradually here continuous diarrhea, and fever, and mainly weight loss. The person is become as too slim. It is also called as slim disease. It is also called as what? Slim disease. So these are AIDS related complex characters. Okay. Then exactly AIDS, when we can call as AIDS means here. So when gradually the number is, the white blood cell number is less than 200 into 10 to the power of 6 in 1 liter of blood. That is actually AIDS character. The AIDS character is what? When the T4 lymphocytes number is less than 200, but actually normal average number is, it is more than 900. More than 900 into 10 to the power of 6 in a 1 liter, right? So this is actually AIDS character, AIDS symptom character. This one is right. And next one is prevention of AIDS, and we know that there is no particular treatment of HIV virus, so that's why curing is better than prevention. Here how we are going to prevent? So by just we have to awareness to the people. We have to give the awareness regarding this HIV as well as AIDS. So further, generally World Health Organization and some of the other than government institute also be work out on to spreading of knowledge regarding HIV as well as AIDS, right? So prevention methods are what? Prevention methods are mainly, first, avoid the multiple sex partners. Okay. Next, like especially in hospitals, like needles, syringes, and those who are addicted to the drug, the intravenous drugs. So we have to avoid that using of needles, and safe disposal of needles. Safe disposal of hospital wastages. Hospital wastages like needles, cloth, blood, like we all are. And one more is here, safe sex by using of condoms. Okay. And also especially in this case, because of in this case innocents are going to suffer from the HIV, that is blood transfusion. Before the organ transplantation as well as blood transfusions, compulsory we have to check. Along with the blood type, we have to check the HIV positive or negative, that we have to compulsory check. Okay. And mostly here avoiding of pregnancy. Here if the mother, it means the disease from mother to fetus through placenta. Okay. So this is actually prevention of HIV. Okay. Next, treatment. So here treatment is mainly the treatment are first what I have to do. We have to inhibit the activity of these enzymes which are present in virus. Inhibit the activity of reverse transcriptase enzyme, integrase enzyme, or protease enzyme. If you successfully inhibit the reverse transcriptase enzyme, there is no possible of formation of double-stranded DNA. If it is not formed, then how is going to integrate it with the genome, right? If we inhibit the integrated enzyme, then the integration with the host genome also be not possible. Then it is not going to replicate it. So if we inhibit the protease enzyme, the glycoprotein molecules are not formed. So during this assemble, like a complete virus particles not going to form. So like such drugs we are going to use it on, right? Treatment is inhibition of enzymes. Virus containing enzymes. Here mainly two treatments are we are using, that is HAART. Second one is cocktail treatment. In first case, here human like active antiretroviruses therapy. Here human active antiretrovirus or reverse transcriptase enzyme therapy, which is going to give the a little result it will give. And second one is cocktail treatment. In cocktail treatment, it is a combination of one or two enzyme inhibitory drug. Enzyme inhibition drugs are we are using, right? So this is regarding treatment. Okay. So this is actually complete session regarding HIV. Just look at the NCERT diagram. Here is given. So this is what HIV virus. Okay, these are the spikes. So this box is done. This complete cell, it is macrophage cell. Macrophage cell. This is complete. The box is actually equal to one macrophage cell, right? Now first it is entered through endocytosis process. It is entered into the cell. During that, this virus containing virus and enzyme molecules are entered into the cell. So this protein, outer coat of the protein, it is shed outer side of the macrophage, right? Now what happened? So this RNA, this is a single stranded 2 RNA molecules. These are the 3 enzymes. Out of that, this one is here, this enzyme is reverse transcriptase enzyme. In the presence of reverse transcriptase enzyme, So this single stranded DNA, it is transcribed to form of DNA. Gradually compromised DNA is become as double stranded DNA is formed, right? Now this double stranded DNA, so this is double stranded DNA again in the presence of integrase enzyme. It is going to incorporated into the host genome. Host is what? Macrophage. So this is a nucleus containing genome. Now this viral DNA, double stranded DNA, it is going to integrated with the host genome, right? Now what happened? Now along with the genome, the viral DNA is also going to get doubled up, and here this DNA is going to produce transcription process. It's going to produce mRNA. So this is what? mRNA, right? So this mRNA is exactly equal to what? A single stranded RNA of virus. Now here in the presence of protease enzyme, this RNA is going to code for some proteins. Out of some proteins is become as enzymes. Now what happened in the cell? Protein molecules are there as well as enzymes these are the enzymes molecules also be there, and number of these RNA molecules also be there. Now all are going to assemble to form of like one provirus is formed. This is provirus. Where it is formed? In the cytoplasm of host cell, nothing but macrophage. Now what happened? Through exocytosis process, these cells are entered into the outer side of the cell. Okay. Then later going to attack to helper T lymphocyte cells, later dendrites, later like it is going to affect lymphatic nodes also going to affect it. And swelling of lymphatic nodes also be one of the main symptom of ARC complex, right? So this is actually how the viral is HIV virus is replicated in within the host. Okay, so this is the entire information regarding HIV. Okay, thank you all of you. In the next class we are going